It's a great pleasure to be here, and I thank you, Alain, for inviting me. I'm not going to talk about morphogens or morphogenes, but about morphogenesis. And like I say, it's great to be here. It's actually great to be a developmental biologist and cell biologist in 2018. And part of the reason for that is the stuff we've been seeing over the last couple of days. We've learned so much about morphogenesis, about development, about cell fate decisions, especially in model organisms, and I'll return to that in a moment, that we really are looking at a completely new stage, I think. And this is illustrated by a story I'll tell you from when I recently went to a developmental biology meeting in Warwick, the BSDB meeting, along with a few others who were here. And when I picked up my rental car at Birmingham Airport, up there you see filthy weather, 7 degrees centigrade, and you see the clerk, typical English, you know, they're always these, you know, chatty chappies, and we chatted. He wanted to know where I was going, and yeah, development, genes, man. And he said, have you made any dinosaurs yet? And so now, 15, 20 years ago, you would have just said, okay, sweetie pie, you've seen Jurassic Park one time too many. But when he asked it, I thought, you know, it's not that crazy. That's what I mean. We've got so much information now, we can actually get there. And I'll tell you that that is my dream. So, but just to step back, it was Erwin Schrödinger in 1944 who asked the question of how the events that take place in space and time within a living organism can be accounted for by physics and chemistry. That's what we've been hearing about the last couple of days. We're getting to the point where we have the data to be able to think about that. And it was a whole 50 years later that Lewis Wolpert asked whether the egg will be computable. So given the total description of the fertilized egg, the total DNA sequence, location of all proteins and RNA, which we don't have yet, whether one can predict how the embryo will develop. And again, we've seen over the last couple of days that we're looking at what individual molecules do, what individual Eric is about, and James, they're both measuring the numbers of molecules and how they function. So I think we're getting there. And so I think the Drosophila embryo presents a particularly good example. You can wonder whether model organisms are useful, and Eric and I had that discussion last night. And the point is that the model organisms, other organisms are as valid, and Drosophila may have been a totally crazy one to use, but we have accumulated so much knowledge, so many tools. We understand in such great detail that I think the Drosophila organism is a good object as an extensively studied paradigm to try and achieve what Schrödinger and Wolpert were postulating. And so my dream is to build an embryo in silico based on physical and biochemical parameters and the genetic rules derived from the work of the people we've been hearing here that can recapitulate gastrulation and make correct predictions as a way of testing whether we really understand everything. And of course, the ultimate dream would be to build an embryo, to use to quote the much overused Feynman quote, what I cannot create, I cannot understand. So, that's just going a little bit overboard. I'm getting old, and these are sort of end-time visions. But like I say, the example, let me just bring this over. The object is the Drosophila embryo and the process of gastrulation, which is shown here in a very old slide, an embryo, a before and after image. Before the embryo gastrulates, it's a single epithelial sheet of identical cells, about 6,000, that, however, express different genes already. So what's shown here is the transcription factor twist, which is expressed in those cells that you see at the bottom panel have gone inside and made the mesodermal germ layer. So we know so much about this embryo. So we know basically all the transcription factors that trigger the events that happen between the top and bottom embryo in the previous slide. So they're shown on the left, and these are the transcription factors. We've heard about some of them that make the first movements in the embryo happen. So the green bands their transcription factor runt, creates dorsal folds, and et cetera, et cetera. So that's work from Eric's lab, and then the red region is the mesoderm that will go inside to make the mesoderm, and we'll hear from Thomas in the next talk a little bit about that blob at the end of the embryo that also undergoes cell shape changes under control of Huckebein and Taylors. So we know the transcription factors that direct these movements, and we actually know a fair amount about the cellular behaviors that are directed under the control of these transcription factors. And the one we know best about is the one in the red domain, both from the lab both from Eric's lab and my lab, and I'll quote some of our jointly generated knowledge in the next few slides. So if you look at a cross-section of the embryo between the left and the right stages, you see that the way the cells move inside is by these cells on the ventral side changing their shape. They constrict on the outer apical side, they shorten, they go inside, and then they fall apart, undergo transition to mesenchyme, and migrate out and divide and form the mesodermal cell layers. And the transcription factors that are expressed in these cells, twist and snail, are in charge of orchestrating all this through deploying a set of downstream genes. And again, through Eric's and my lab, we've learned what all of these downstream genes are doing, and they basically act on a set of subcellular signaling molecules that end up modifying the adherence junctions that hold the cells together, and the actin cytoskeleton, that is the active component in generating shape. And here's from Adam Martin a little diagram. What happens is that these cells on their outer apical side elaborate a cross-linked actin myosin meshwork that gradually constricts and shrinks the cell surface of those cells. So the dogma about this event is that the invagination of these ventral cells is determined exclusively by the activities within the cell. So it's probably cell autonomous, but certainly regional autonomous. And the evidence, again, is some very old stuff, and again shows Eric's and my parallel activities always. This are experiments I did with Siegfried Roth when he was a postdoc in Trudy Schubach's lab, and I was hanging out in Eric's lab for a while, where we transplanted nuclei into an embryo. You see at the top is an embryo that doesn't have a ventral furrow. It doesn't have twist all over. That's because it's mutant, and it can't make a ventral furrow. But if you transplant wild-type nuclei into this animal, then those nuclei will make their own little furrow. So they can do it without any help from their surrounding. And you can go to more extreme situations where the entire embryo is changed in fate, and you can put in some cytoplasm that is autonomously able to make a furrow, and it'll make a furrow anywhere you put it. So there's nothing special about the egg shape. You can make the furrow go across rather than lengthwise, and if you make a round spot, it'll make a crater instead of a long furrow. So that's all the data that says these cells know what they're supposed to do on their own and do it on their own no matter where you place them. But these cells are actually part of an entire embryo, and so they somehow have to get the rest of the embryo to participate in their activities, because if they didn't, if they just constricted and the rest of the cells just sat there, that would rip the embryo apart. Why is this not moving? Oh, this is weird. Stuck. Sorry about that. Okay, escape. Okay, let's just move on to the next one. Okay, so we understand what these individual cells do, but to understand how the whole embryo actually generates the shape, we need to understand how these cells interact with the rest of the embryo. Even if the rest isn't actively contributing, it has to comply. And so that's what we've been trying to find out over the last few years. How does the rest of the embryo actually participate in the activities that allow the mesoderm to invaginate? And there are two extreme models you could have, two extreme scenarios. You could imagine that as the ventral furrow folds in on the ventral side, the rest of the embryo just behaves like a rubber ball, like a balloon, like an elastic sheet. So everything just gets stretched as these cells go in. You could do that in a balloon. You can poke in, and then the rest stretches. The alternative is that all the different cells have their own little program. So it's like a big ballet, and the choreographer has told them all what to do. This is not such a mad idea as it might seem. The first is obviously simpler, because we know that at this stage, the cells already have different fates. Eric has shown you all the beautiful anterior-posterior patterning, but also along the dorsoventral axis, a lot of patterning has already gone on, as this beautiful slide from Angela Stathopoulos shows. And by the way, while the AP axis may not look so much like a neural tube, this looks a little bit more like James's neural tubes. So the embryo is actually already subdivided into lots of different little cell fates. But what we decided is that if we wanted to figure out how this choreography happens, we'd better be able to observe in vivo what each cell is doing at each time point. And that's not so easy, because the process that I showed you in the drawings happens extremely quickly. It's basically over in 20 minutes. And so the cells move very fast. They undergo their cell shape changes very fast. And we wanted to see all cells at once, which is not easy. So this is a project that was started by Matteo Rauzzi when he joined the lab quite a long time ago. He now has his own lab at the University of Nice. And he initially started building his own microscopes, but he then ended up collaborating with Lars Hufnagel's lab, who built a very nice microscope based on single plane illumination, and which uses 2 lasers and 2 cameras. So you can collect data very fast and at reasonable depth. And what this collaboration resulted in, and lots of tweaking, lots of optimizing, is Matteo being able to collect entire data sets of embryos throughout the whole depth. And here we're focusing through in silico, a reassembled embryo from anterior, posterior, dorsal, ventral, side to side. And he was able to collect data sets of this type at 20-second intervals. Now, there's masses of data in here, far too much for a brain, at least my brain, to handle. And so what we decided to do is to extract, for a start, only the data on the surface, because that's where the action is. That's where the greatest cell shape changes happen. And so what Matteo and his colleagues in Lars's lab did was to write an algorithm that extracts only the surface data and then rolls them out like a world map, except it goes north-south instead of east-west. And so we'll observe now such a sheet of cells which represent the entire embryo gastrulating, and the furrow will be in the middle. There will be 2 dorsal sides because we go more than once round. So here, oops, here we don't go. Here we go. So you see in the middle, you'll see the furrow appearing. You'll also see these other movements that I mentioned. The head fold is appearing close to the head now, and you'll see the posterior midgut coming around the end of the embryo, bringing with it the pole cells, the future germ cells. And in the cross-section, you just see one plane that was taken right through the middle where you see furrow formation. And as you can see, it's a very rapid process. And we can now really see what does every single cell in the embryo do? How does it respond to, or how does it participate in the activities that the ventral furrow undergoes? And a slightly easier graphic way of looking at this is to make a chymograph, and I'll use this chymograph to show you a couple of findings in a moment. But another way of looking at it is actually to make measurements of the cells throughout the embryo. You can measure their speed, their eccentricity, their directions of movement, etc. And so let's talk about some of those data. So what Matteo did was to subdivide the embryo into cell groups and measure their behavior. And so in this diagram, what we show is the eccentricity of the cells, and that's shown in color. So a hexagonal or round cell is shown in blue, and an isometric cell is shown in red. So a stretched cell will be red. And on the y-axis, we show the direction of anisotropy. So a cell that is stretched along the anterior-posterior axis is at 0 degree, and a cell that is stretched across along the dorsoventral or right-left axis is shown in red. And so these are the dorsal cells, and you see that initially they don't do anything at all, and then they become very stretched, and the direction in which they stretch is side to side. So that's one population. What do the other populations do? At the bottom, you see the ventral cells. We already know what they do. They narrow their surface, and they become very wedge-shaped. So initially, they also start out round or hexagonal, and then they become stretched, and you see they become stretched in the different direction, namely anterior-posterior, and then they disappear. We can't observe them anymore because they dive into the embryo. Meanwhile, the cells in the middle just stay pretty much round. Now, what you also see is that the dorsal cells that I talked to you about actually stretch long after the ventral furrow has gone in. And if you look at that in the kymograph, you see clearly that there are two phases. So initially nothing happens, cells all stay in their place. And then in the middle of the embryo, you see the cells converging to the center and disappearing. That's the constriction of the ventral cells. But at this time, the cells on the side don't do anything at all. It's only later that they move towards the middle. And it's only at that stage that the dorsal cells stretch. And there's something funny has happened again, I see here. So we have two phases then, and we have different cells doing different things. The lateral cells just sit there for a while and do nothing, while the ventral cells dive in. That's very strange. Let me see. Have I skipped a slide? No. So okay. So never mind. So what we ask then is, are these behaviors, this order of behaviors, and this differential behavior of cells, does it matter? And what you also don't see here very well, and that's the slide that's missing, is that if the ventral cells constrict and go in, and the lateral cells don't move, something has to give. And what gives are the cells that sit in between. There's 3 rows of cells on each side that become extremely stretched. So it seems as if these cells have different behaviors. One of them just stays round and doesn't budge. One of them stretches, and others constrict. So does it matter? And one thing we did is use a technique that Matteo developed where he can fix cells. So if you zap the surface of the embryo with a laser, what happens is that coagulates the cell with the overlying vitelline membrane, so the eggshell, and it attaches the cells to the outside so they can't move. And so what he did in this movie here is to do this to cells on the side. And what you see then is that the furrow begins to form, but it can't pull the tissue in towards the ventral side, and then the furrow doesn't, it just gives up. Actually, the cells divide and everything goes haywire. So, and what you see at the beginning is that the cells immediately adjacent to the furrow are able to stretch, but what's clear is that the further out cells are unable to stretch. So this says, indeed, that these cells have different properties, which we've also then measured. So what we have is a central population of constricting cells neighbored by 2 or 3 rows of soft cells that respond, and then 2 cohorts of very stiff cells that aren't able to stretch. And then their movement, in contrast, is then again accommodated by stretching of soft cells on the dorsal side. So we have different mechanical properties of these cells, and we've actually measured this. There are very great differences in tension across these cells. that apparently determine the behavior of the embryo at this phase. Now, we then became interested in the underlying mechanics, the underlying causes of this, and the obvious candidate for being responsible for these mechanical differences of the cells is the actomyosin meshwork, which we already know is doing the work on the ventral side, and we know from Thomas and our and other people's work that the apical cortical actomyosin meshwork determines the mechanical properties of these cells. So, and indeed, actomyosin meshworks are the main driver of morphogenic activity. Here's cells during convergent extension in Xenopus laevis, and you see this amazing pulsating actomyosin meshwork. And here again from Adam's work, Adam Martin's work, is the actomyosin meshwork in the ventral furrow. Again, you see these pulses that mediate the morphogenetic work at this stage. And as I mentioned, the cortical meshwork in the Drosophila embryo also changes over time. So this again from Matteo. Before gastrulation begins, there's a sort of diffuse actomyosin meshwork at the surface of all cells, and it's equal in all cells. But as gastrulation begins, this meshwork becomes differentiated among the different cells of the embryo. So I'll now talk about some theoretical modeling and simulations that we've conducted with the aim of being able to understand these meshworks better. And this is the work of Giulio Belmonte, a postdoc who was a joint postdoc between François Nedelec's lab and mine, whose aim was to understand precisely, to model and understand the behavior of actomyosin meshworks. And what he proposed to do was to take these cell populations, make simulations for each type of cell and the way the actomyosin meshwork behaves in each type of cell, and then ask how do such cells in silico interact with each other, and eventually build this entire carpet of cells that we've seen and understand it. And to do that, he used a simulation that François Nedelec had developed, and which uses filaments, polar filaments with a plus and a minus end that are able to bend with an elasticity that you can determine. It uses connectors which can diffuse, shown here as AB, so cross-linkers, which can have on rates, which can cross-link, and which have off rates. And if they're motors, if you program them as motors, they can move either to the plus end or the minus end, and they come off. Anything can come off, either when it comes to the end of the filament or with any off rate you would like to determine. And you can choose any parameters of these you wish. You can mimic known biochemical parameters. And as I said, that's one of the nice things being alive in this age where we do know so much about the individual motors, about molecules that constitute such networks. So what Giulio did was to take this model, which consists of filaments, motors, and cross-linkers, or which you can program to consist of filaments, motors, and meshworks, and add to it membranes, shown here in blue, with adherence junctions, which are basically just a connector. And he also included vertices that hold the membranes together. And then just ran a simulation, and it was very pleasing to see that this looks very much like what we see in the ventral furrow, until at some point it stops looking like that, because the actomyosin meshwork just collapses in the middle and rips off the membranes. That does not do any morphogenetic work. And it became clear that we actually, and we played around with this and put in neighboring cells with different properties, but eventually became clear that we don't really understand fully how a simple meshwork itself. What are the rules that govern the behavior of such a meshwork? And so Giulio went back to the cell-free system, just filaments, cross-linkers, and motors, and explored how the whole parameter space of how such mixtures can behave. And so here's an example where he took 2,000 actin fibers and titrated in varying concentration of cross-linkers and varying concentrations of motors. Now, if I asked you to predict what would happen, I don't know, anybody want to predict? Some of you have seen it. You can guess. You would probably not predict what happened. What happened was this. So he did then a simulation for each of these little squares, and what the next slide shows is the outcome of the simulation after X minutes. So you can program time in this. And what you find is that the bottom right is not surprising. If you have lots of cross-linkers, the meshwork just freezes. It just stalls. It just sits there. What's more surprising is the top left corner. If you titrate myosin into this meshwork at very low concentrations or zero cross-linker concentrations, it never constricts. So myosin and actin alone do not contract. So that was a surprise. And then it's only at certain ratios of motors to cross-linkers that such a meshwork will contract. Actually, that was very nice because this predicts an experiment that had been done previously, which was done in vitro, which did exactly the same thing. Filaments, stabilized filaments, and varying concentrations of myosin and alpha-actinin. And you see there are many conditions where you get no contraction, and those are the circles. But in a certain domain, you get contractions, very similar to the predicted results. So we played around with that, but then we began to think, well, if, you guys, had you thought deeply about this, we should have been able to predict this because we know everything about these filaments. We know how they bend, we know the on-off rates of the motors and the cross-linkers we put in. It ought to be possible to predict. So we then decided it would be worth thinking about, can one predict, or how can one predict how a meshwork, a connected meshwork with active motors in it, random meshwork, under what conditions will it contract? Now, there had been theories about that already by a number of people, but usually for very small, some of them here in Paris, mostly just for very small meshwork, sometimes 2 filaments, sometimes, a small number. All of these theories looked at filaments, 2 or a few, and the molecules that cross-linked them. François, when he was thinking about this, one day decided to turn this around and say, let's not look at 2 filaments and what they do to each other. Let's look at 1 filament that is connected with 2 connectors to a surrounding meshwork and ask what that filament and these connectors do to the meshwork. And to do that, he said we should look at the distance between these 2 connectors within the meshwork and ask what happens to that distance, depending on what the connectors do. So this is a more simplified form. So on the left, if these 2 connectors are, let's say, alpha-actinin, so they're just binding proteins, then nothing will happen to the distance between them because they don't move. If they move towards each other, for instance, a motor and a connector, then they will shorten the distance A, and they will actually be pulling on the network, so they'll give a local contraction force. If they move away from each other, a different end motor or a different orientation of the 2 molecules, then they'll push on the network and make a local expansion force. This latter is only true if the filament is actually a stiff rod. If it's flexible, then what happens? It can buckle. And then some of the pushing force, maybe not all, depends on the distance, etc., and on the flexibility, some of the pushing force will be lost. It'll be spoiled. So now what you can do is you can just say these are the only possibilities. And so depending on the types of connectors and the flexibility of the filament, we ought to be able to predict what happens. So let's look at that for a simple system, actomyosin, a plus end-directed motor, and a cross-linker. And if you have equal numbers of these in the system, then you have 4 possibilities for their sitting on a filament, and therefore 4 possible outcomes for their behavior. If they're both just binders, then the distance between them, so length A, over time doesn't change. If they're both motors, the distance also doesn't change because the motors just both walk along. It's only if you have them moving towards each other at V, the speed of the motor, or away from each other, that you have a change. You have pulling or pushing, as I explained in the last slide. And then all you have to do is add up all the possibilities in the system and add up all the effects. So the two first are static or neutral, and then you have a pulling and a pushing, but they cancel each other out because they're same frequency, same speed, so you should get no change in the overall shape of the meshwork if you have rigid filaments. If the filaments are flexible, then the bottom condition, you lose some of the plus V because the filament buckles. So therefore, pulling should dominate, and you should get contraction. And so that's all very well. That's a prediction. But hey, how do we figure out whether it's true? That's where we went back to Giulio's model, to Giulio's simulations, and we just put all of these conditions exactly with, in this case, with actomyosin conditions, with myosin and alpha-actinin conditions in, and looked what happened. And what you see is, with rigid filaments, you see them all wiggling around. But overall, the area that the meshwork occupies doesn't change. But as soon as you make the filaments flexible, boom, it contracts. And here's a very high resolution of the right meshwork. And you see how over time, we just randomly colored in a few filaments. They bend and buckle, and everything converges towards the middle. So that's for one type of, or actually for two types of connectors. In theory, you can take all sorts of activities and combine them ad lib in pairwise connectors. Most of all, well, many of those probably don't exist in nature, some do. And then you can make predictions what they should do to a meshwork. And here's a matrix of such predictions where an empty circle means nothing will happen. Green arrows to the inside means contraction, and red means expansion. And you then can go back again and model this. And here's this model, bang, it contracts. And in fact, every single circle in the simulation behaves exactly as the theory predicts. So that's nice. So we have a theory. If we make the filaments flexible, everybody contracts, and again, that happens. So you can expand this. This is for one type of connector. You can now combine the combinations and get huge matrices for stiff. I mean, the parameter space is almost infinite, but we just wanted to at least try a few more models. And so we made all these predictions, and then we did all the simulations, and we developed a way of quantifying the extent of, or the speed of contraction and expansion, both in the simulation and in the model, and then compare them. And you see here, we plot the prediction against the simulation, and they come out very well. So it seems that this theory for how such a meshwork behaves works well and does, in fact, predict reliably. So those are all very theoretical meshworks, but in fact, the theory can be used to describe known situations, not just the meshworks I've just showed you. So a very well-known example is the muscle fiber, the sarcomeric unit, which can be, which can be represented in the theory and behaves accordingly. You have buckling-dependent contraction that's been shown in vitro on the left here. That, too, we have already seen is a mechanism that drives contraction. And then at the bottom, we have a sort of a mechanism that creates asters of microtubules in this case, which is caused by end dwelling of plus-directed motors. Now, the bottom mechanism has been described for microtubules and was thought to function only for microtubules. But recently, the group of Gijsje Konderink in Holland has discovered that it also, at least in vitro, occurs for actin filaments. So what Gijsje and her grad student Victoria Walrab did was make actin filaments in vitro and add myosin minifilaments. And you see here at the top that these myosins will land on an actin filament, will move towards the end, and then they'll sit there for a while. And this leads to what they call polarity sorting. So if this myosin sitting on one filament then attaches to another filament, it can walk along that one also to the end, so it connects the end. And if it does that throughout the meshwork, what you see, obviously, it'll create these asters. And so that's what they observed in meshworks, in artificial meshworks, which they constructed out of skeletal actin and myosin. So that's a little bit of difference from cellular actin myosin. But that's what they observed. And so they asked us, what would you predict? And in fact, they asked us what we would predict after they'd done the experiment already, but that's fine. What they observed is that the way in which these meshworks behave depends on the density of, and again, the relative concentrations of myosin and actin. Now, there's no alpha-actinin in here, so I'll return to that in a moment. You should have noticed that. These are meshworks that contract without added alpha-actinin. So what they see is that at different concentrations of myosin, which they show here, they get very different behaviors. The middle one is amazing. It just contracts into one big aster. The left one contracts into several small spots. It's shown diagrammatically above. And the right one is completely stalled. So this, of course, should have bothered us and did bother us because we've said previously there is no contraction with myosin and actin only. But the myosin that we had in our simulations had the behavior of when it reaches the end of the filament, it immediately falls off. And so just to reiterate, if we model actin and myosin alone without end-dwelling, hang on, where does it go here? You see this behavior. They all slither around past each other, and they don't contract. So when Giulio then added end-dwelling, what he saw in the model is this. So boom, exactly what they observe in vitro. And again, you can do this for a large phase diagram. And what this shows is on the right, certain set of conditions without end-dwelling, and on the left, conditions with end-dwelling. And you see that without end-dwelling, you have an area where the filament buckling does create contraction, but not in others. Both networks stall at high concentrations of alpha-actinin in this case, but at conditions where buckling alone would not give contraction, this end-dwelling can. Whether that is also true for cellular actin remains to be, and myosin remains to be seen, and that's something we'll have to be looking at, and they will be looking at in future. Okay, now there's one thing about all these meshworks that you may have noticed, and that is that they don't resemble this, because they all contract and collapse in the middle of the field. Whereas in vivo, you get these pulses, these continuing pulses. But there's also, and again, here it is. So, but there's also one thing that we've left out of our system, and that is in everything I've shown you so far, we have stabilized actin filaments. Whereas, in fact, in vivo, what happens is that we have actin turnover. And so when Giulio added actin turnover to the system, this is what he got. Again, this is a particular condition. It's periodic boundary conditions, and it's a certain rate of turnover. The turnover here is not treadmilling. It's just take out a filament and put in another one at random positions. But you do get pulses. So that, again, is interesting. It says that pulses as such don't need any external trigger. They are an inherent property of actomyosin meshworks under certain conditions. I mean, in certain parts of parameter space. So this is what it looks like. And we then decided to figure out, when do you get pulses? What are the conditions to get pulses? And we first returned to a refined model where we have a field with or without a boundary, so a cell outline, and where we just explore parameters. Now, I think in this one, if you just have an open field in which you will now have random taking out and putting in of actin filaments, what happens is, oops, sorry, you do need to see what happens, is everything still converges to the middle. It's more lively, there is some dynamic in there, but it all collapses into the middle. So what we then did was to add membranes again. The rest is just the same system. We use membranes to which the filaments can attach, and we have the turnover. And then we explore different parameters. So here is the one you saw before. What happens if we add a membrane? Does this work? Yes. You begin to see foci forming, and the foci float to the side of the cell and then attach. But this does continue over time. So it looks a little bit more. It depends, like the real thing. Oh, the time's cut off at the bottom. If you change the timing to either very short, either very, very short half-lives, 5 seconds, you see you never get foci forming. They try, but then they dissipate. And if you have very long half-lives, then the foci form and float to the side of the membrane where they get attached, and nothing happens in the middle again. So there clearly is a temporal component. And if you measure the center of the field, which is the way some people look at these contractile pulses, if you measure the apical amount of actin, you see you get very different behaviors. In black, at very slow turnover, you do get oscillations, but nothing ever goes away completely, and you don't get high peaks. At very long half-lives, the bottom one, most of the meshwork is depleted from the center, and you get you give pulses that persist, but then disappear completely. And it's only in the middle two conditions, 40 and 80 seconds, that you get some kind of pulses. And there seems to be a frequency dependent on concentration. Now, the trouble is, nobody actually knows what they mean by pulses. And it's actually also not so easy. So if you look at this, how many foci are there? Are they pulsing? Are they just floating about? Actually, even in vivo, we don't know what pulses are. We don't know whether there's turnover within them, whether they float around, whether movement from one place to another is reassembly or actual movement. And so we decided to try and come up with a way of quantifying and measuring this. What we're doing currently is subdividing this in a grid and then just counting the number of actin molecules in each grid, and then you can false color this, create a heat map, and then you can say every time you're above a certain point, you call it a focus. You can also distinguish them. You can color them and then ask, oops, why does, oh, maybe it does. No, it doesn't run, it does. So. There. And you can then say, you can now see that they often merge. They sometimes, rarely, if ever, disappear, and they float around. And you can now set your thresholds and look at that behavior. And what you see is that foci come and go. We do not see any periodicity. It appears to be stochastic. And most of the foci, what they do is they merge and go to the edge of the field. Very few disappear. Actually, here it's plotted. So this is just over time, the number of foci, just cumulative. And you see that most of them merge, very few of them disappear. So we can now use this. There's a lot to be explored about the behavior of these foci and these pulses. There's a lot to be done with measuring in real life what these foci actually are and how they behave. But perhaps with this, we're getting to the point where we can ask, have you built any embryos yet? Like I said, that's my dream, but that's for the future. And this is just to thank the people who worked on this. Matteo Rauzzi did the imaging in the first part. Giulio did the simulations, and together with Francois, we did the theory. And lots of help from Lars Hufnagel and other people at EMBL and outside, and at the top right are the foundations. Thank you.